Okay. Hello, everyone. I'm Jeremiah. I'm an MD PhD student here at UT in the lab of Neil Hayes, and I'm just going to talk a bit about my work on this project you've heard so much from already, involving these tumor models. So you heard enough about breast cancer, but what I would like to emphasize is that there are subtypes of breast cancer, and these are typically defined by the expression of either hormone receptors, such as estrogen and progesterone, or a growth factor receptor called HER2. When you lack these, you're referred to as triple negative breast cancer or TNBCs. And the important thing about TNBCs is that they lack targeted treatments because they lack these receptors. So because of this lack of targeted treatments, as well as racial disparities, TNBC has the worst outcomes. So one thing I don't think was mentioned very much was we actually see more cases of TNBC in African-American populations, and the mortality is higher in that population. So I think a lot of this has been attributed to, systemic racism, but there could be some genetic components that drive this population to get this more aggressive disease. So while triple negative breast cancer is a subtype of breast cancer, triple negative breast cancer also has more subtypes on top of that. So using gene expression profiling in the 2000s, the original 5 subtypes of breast cancer or molecular subtypes were defined. One of these includes triple negative breast cancer and has been called basal-like as well. And then some groups later, also using gene expression profiling, were able to identify more subtypes of basal-like or triple negative breast cancer. So you'll notice that these subtypes are defined by the expression of certain biological pathways, such as immune signaling, differentiation and epithelial-mesenchymal transition, as well as androgen receptor expression and cell cycle control and DNA repair. I think you guys understand how this model works, so I'm not going to spend too much time on this slide, but in general, the males get prostate cancer, and interest to us, the females get hormone receptor negative breast cancer. Okay, so here is a heat map showing the results for the RNA sequencing. Each column represents an individual mouse, and each row is a gene. So in this big box down here, I've got about 4,500 genes, which represents the top 25% of the most variable protein coding genes. At the top, I've also included a zoomed in view of a few genes, and they're mostly just the hormone receptors, such as estrogen 1 and 2, progesterone, HER2, the growth factor receptor, and then the androgen receptor. I've also included the SV40 transgene itself, because as you know, 2 of these strains did not develop cancer. So one thing we were really worried about was, oh no, this transgene is just not being expressed at all in these cells or these strains. But it turns out that transgene is being expressed just at a very, very low level, especially compared to the rest. You can see how blue it is. So in general, these samples are normal adipose tissue, so they look much different than the rest of these tumors. However, there is a group of mice that are clustering with these normal tissues, so these could be deemed maybe normal-like, but my hypothesis is that they just have a higher proportion of normal contamination. So what I mean by normal contamination is stromal cells invading the tumor, or maybe just when the tumor was resected, they grabbed some of the surrounding normal cells. So that's something we see often in clinical practice. The next thing you'll notice is that over here on the right, there's a pretty well-defined cluster that has very high expression of the SV40 transgene and a gene expression pattern that is much different from the rest. So there could be something about these strains that make it more likely to express this transgene at a higher level and thus have a different biology. So this is the same heat map, but I've split everything by strain. And you'll notice again on the right that the normal samples, not much variability. This is something that we often see because in normal tissue, gene expression is very tightly regulated and it's cell specific. However, if you look in the all the mice from all the different strains, you'll see that not only is there quite a bit of variability across the strains, but some of these strains actually have considerable variability within them. So if you look at strains like this light green one here, you'll see that half of the tumors have one phenotype and the other have another. There are some strains that have all the same tumors. So this could mean that maybe there are some strains that have certain heritable factors that make it more likely to look like one tumor of the X. To me, this emphasizes that sometimes it's not best to collapse these to strain means because there is so much variability, you might lose out on some of the nuance there. So for the purposes of this project, I keep them separate and treat them like individual patients or tumors. So next we removed the normal specimens and just kept the tumors. And then we did a hierarchical clustering approach, which identified 4 statistically significant clusters. So right off the bat, you can see here on the heat map of the clusters as well on the right, I have a PCA plot that this blue cluster, or cluster 3 as it's arbitrarily named, is primarily driven by the expression of the KRAS oncogene. So you can see that here in the heat map, most of these samples have very, very high expression of KRAS. My hypothesis is that there is some sort of structural alteration here, probably a chromosome level or arm level, or even just gene level amplification event. So that's something that we would like to look into with the whole exome sequencing. Interestingly, with that KRAS overexpression, you see very high expression of the SP40 transgene. So there could be some sort of link there that is driving both of these at the same time. You can also see in red here, it's a small cluster, there's only 5 samples, but they overwhelmingly have the highest expression of the AR gene. So these could be a group of samples that are driven by the overexpression of the androgen receptor and the androgen hormone. The other 2 classes, the main one is this purple one, and the green one in the middle looks something like in between the purple and the blue. And you can really see that here on the PCA plot as well. So I'm showing the same heat map, but again, split by strain. And you can tell that, at least when you look at this table, there are a few strains that have a very high correlation with being placed into a single cluster. So this blue cluster, which was the one with the high KRAS, it seems like there's a few strains that have a very high rate of being placed into this class. If you look on the PCA plot, you'll see that this is a fairly well-defined group. And then when you look at the purple class, you'll see that, again, they are separate, but this one is a little more variable. And then again, there's this class in the middle. And so the interesting thing is when one of these strains have a tumor that falls within another class, more often than not, it seems like it is being placed into this green class, which could be something, again, looks like in between the other 2 classes, at least when you put them on a PCA plot like this. So again, there could be some genetic factors contributing to more or less placing you in one gene expression class versus another. Okay, so yeah, this is great. We found some gene expression patterns, and they're statistically significant, but none of that really matters unless you can find some biological correlations that go with it. So here, I'm showing a heat map again, but instead of genes here in the rows, I'm showing gene sets. So I do actually include a few genes here at the top that are differentially expressed between this blue class and this purple class. And these are kind of just placeholders for some of these classes, or for some of these gene sets down here. Excuse me. So another annotation track I added here is called TNBC type. So this was using a web tool from the folks who developed the initial 6 TNBC subtyping predictor. And so I applied the mouse data and used this predictor and was able to generate subtype calls from a human predictor on these, on most of these murine samples. So right off the bat, you can see in this blue class here, we have a lot of enrichment for this immune modulatory TNBC type in red. We also have quite a few basal-like tumors here in black, and then over transversely, or conversely, sorry, in the purple class, you see much more enrichment for this mesenchymal or mesenchymal stem-like phenotype. So it's interesting to be able to recapitulate these human phenotypes in this really powerful mouse model. So yeah, this first class that I've been talking a lot about with the high KRAS expression is definitely enriched for pathways related to KRAS signaling, as well as immune activation. interferon signaling, and other aggressive tumor characteristics such as epithelial mesenchymal transition and angiogenesis. You can see that this green class also shares some of the phenotypic features of this blue class. However, it lacks the elevated expression of KRAS. So there may be something else driving these tumors in a similar fashion. You also see enrichment in 2 of these classes for proliferation markers, cell cycle progression, and DNA repair. So those are typical tumor hallmark characteristics. So having those differentially expressed in the classes is somewhat interesting. So on top of this, you can see that this purple class, while it is not driven by KRAS signaling, it seems to be more dependent on growth factor signaling, such as EGFR, FGFR3, and it's also enriched for differentiation or epithelial differentiation markers such as TP63 and SOX9. So this class could maybe represent one that is a more differentiated tumor, but is still experiencing this high proliferation phenotype. So you saw some of the immune results already, but this time I've added my annotations for my gene expression classes. So overall, it seems like the high KRAS class, as well as the other mesenchymal class, seems to have higher enrichment for tumor immune microenvironment. So basically, most of these immune cells seem to be more enriched in these tumors, especially lymphocytes, and as to importance for immunotherapy, CD8 positive T cells, as well as other tumor-associated immunity cell types. In the purple class, it seems like there's more monocyte invasion. I'm not really sure how to interpret that. I'm not an immunologist, but it seems like that could be an important difference between the two. And then there could be strain-specific genetic factors that are contributing to both these phenotypes. So then again, like earlier, I said I don't really like to collapse to strain for this analysis, but when you do, maybe that will allow to identify, certain strains that are associated with things like tumor immune microenvironment, as well as a potential response to immunotherapy. So to wrap things up, basically we identified distinct groups of tumors based on gene expression profiling in this murine TNBC model. These associated with BXD strain, tumor biological pathways, and human TNBC subtypes. Where we're going to go forward with this, I think the lowest hanging fruit is to integrate some of this phenotype data you saw before. So on the right here, I've got an eigencore plot, if anybody knows what that means, but essentially we are relating some of those principal components that I've shown before with different phenotype information. So I included some gene expression here as well, but overall from just this preliminary result, you'll see principal component 1, which represents about like a quarter of all the variation across all these tumors, is heavily influenced by the SV40 expression. It's also heavily correlated with KRAS, and then almost all of the phenotype markers we have seem to go with this. So there could be, I think there definitely should be some more analysis with this to try to integrate my gene expression classes with some of the other phenotype data points that we have. On top of that, we are doing low-pass whole genome sequencing, or actually we have already done that. So it'd be nice to integrate some copy number changes, like I mentioned before, as well as any potential surgical variants, or even if we can do mutation calls, tumor mutational burden, and things like that. So with that, I will call it quits and thank Liza for letting me analyze this data, and to everybody who's worked so hard on this project, and then my mentor, Dr. Hayes.